Okay, everybody. To this point, we've been talking about cellular energy. And remember, the main currency of cellular energy, I like to think of it as sort of the dollar bill of the cell, is ATP. So let's connect this to our biochemistry unit and remind ourselves that chemistry, right, is really about the atoms and the bonds, and mostly about the bonds. If you're making bonds, you're storing energy. If you're breaking bonds, you're releasing energy. The problem is that not all bonds are created equal, right? Some bonds are um, like touching electricity, right? They're dangerous to the cell. So if you were to just break glucose bonds inside of a cell, the energy given off is actually too much for the cell. So we need to convert that energy into ATP. I like to think of glucose as sort of being like $100 bill. Imagine, right, that you wanted to go to the vending machine and get something out of the vending machine. Well, you've got plenty of money, your $100 bill, but you go to put that in the vending machine and it only accepts dollar bills. So somehow, right, you have to convert this $100, which is very useful, nice to have, into something though that can actually be useful for you. And that's kind of what goes on in cellular respiration. It's the conversion process that takes the glucose, the $100 bill, and turns it into the dollar bills of ATP. So we're going to talk about kind of how that happens. So just an overview of the whole picture, right? If you think about it, you eat food, right? And there's lots of different macromolecules in there, and in those macromolecules, there's lots of bonds. Ultimately, you need to break those bonds down into ATP, and then that ATP can be used for all of these things in the lower left, right? Maintaining your body temperature, making your muscles work, carrying out other chemical reactions, repairing cells, all the things that a cell needs to do and needs energy to be able to do. It needs to be able to get that from this process. So a very important process, cellularly speaking. So again, just sort of another overview. You're looking at a process that releases energy in the presence of oxygen. So in other words, you need oxygen to make this work. So if you imagine taking a bite out of an apple, we know from our food lab that we have both starches and glucose in that apple. We have enzymes in our mouth called amylase that start to break it down. We mechanically digest it by chewing it. Ultimately, we end up with a lot of glucose, right, in our intestines. That gets out into our bloodstream, gets out to our cells, and in the presence of oxygen, and in conjunction with the mitochondria inside the cell, that glucose is converted into ATP. And then that ATP, we will learn all the different things that it can do for us. So the overall chemical reaction here should look familiar to you. If you were to take away these 36 ATP, essentially we are looking at photosynthesis reaction, but in reverse. Okay, so cellular respiration has three stages to it. So in order to convert this $100 bill to a bunch of dollar bills, right, we have to go through several stages. So the glucose has to move through a process called glycolysis. Use your root words, glyc, sugar, right, glycemic index, you've probably heard. Lysis, to split, think the lysosome, lysol, right? Cytolysis, hydrolysis. So literally to split sugar. The Krebs cycle, right? Lots of PhDs to figure that out. So we're just going to do an overview of it. And then the electron transport chain. There's going to be a lot going on in these next few slides you're going to start taking notes on, but I want you to pay particular attention to these things. What is happening when it starts? And when it's in particular, when glycolysis starts, what are the inputs, chemically speaking, right? What do we need to make that happen? What's going in? What is coming out of each of the processes? So at the end of glycolysis, right, what comes out of it? At the end of the Krebs cycle, what comes out of it, chemically speaking? Where is it happening, right? The glycolysis is taking place in the cytoplasm. The Krebs cycle, for instance, is taking place in the mitochondria. So where physically is it happening in the cell? And then finally, is it anaerobic or aerobic, and we'll get into what that means. So try not to get overly confused. There's a lot going on here. We don't expect you to know all of it. These are the things to focus on. Aerobic, as a reminder, most of us probably know this, pertaining to air. Really what it means is oxygen is present. Anaerobic are processes that can take place without oxygen. So here we go, page 22, I believe, of your interactive notebook. You should be taking some notes here on this. Your first stage is glycolysis, and it literally means to break sugar. So one glucose, right, chemically is going in, right, is in the cytoplasm. Lots of enzymes involved here. And ultimately, what comes out of it is pyruvic acid. Now, this is an anaerobic process. Okay, so in other words, no oxygen is needed here. So oxygen isn't going into this process. And it takes place in the cytoplasm, right, near the mitochondria. So the things you need to know: what's going in, glucose; what's coming out, pyruvic acid; where is it happening, in the cytoplasm; and is it aerobic or anaerobic? And in this case, it's anaerobic. Okay, so now that pyruvic acid, we're going to get into here in just a minute, goes into the next stage, the Krebs cycle. But just to point out a couple things about glycolysis, this is a very fast process. So if you need ATP right now, right, it's good. But it's inefficient. That's one of the issues with it. So even though it can happen fast, it's not a very good use of your currency. So imagine taking that $100 bill we were talking about earlier, and you gotta have a candy bar. And so you go to your buddy and you say, hey, can you make change for me? And they say, well, I only got $50 in ones. And you say, you know what, I'll take those $50 in ones because I really want that candy. So you're going to take a $50 loss, essentially, right? And you're not getting your full conversion because of how much you want your candy. And that's kind of what it is for the cell. The cell really needs that ATP energy. It's willing to do it inefficiently through glycolysis. Okay, second cycle is the Krebs cycle, or sometimes referred to as a citric acid cycle. And again, this is very complex. A lot of people have spent their entire careers and PhDs learning this, so don't get overly worked up about it. Note that when oxygen is present, that pyruvic acid passes into this Krebs cycle stage. Okay, this is where carbon dioxide is coming out of cellular respiration. So what's going in, right? Oxygen, pyruvic acid. What's coming out? Carbon dioxide. Where is it taking place? In the mitochondria now. So the powerhouse of the cell is where this is taking place. Okay, and you should be able to answer for yourself, aerobic or anaerobic. So again, kind of a overview, a lot of stuff going on in this simplified version, but you can see here's the pyruvic acid going in, here's some CO2 coming out, here's some more CO2 coming out, but also a fair amount of ATP. So this process is slower because it has to go through it is taking place in the matrix, sort of in the membranes inside the mitochondria. So this is a slower process because there's a lot of things going on, but it's more efficient, right? You start to gain a little more ATP, but most importantly, you get a bunch of high energy electrons. Those are important because they drive the final process of cellular respiration. And that is often referred to as the ETC, or the electron transport chain. And really what's going on here, pretty complex, but you're looking at a membrane here, this gray thing, right? And there's an inside of the membrane and an outside of the membrane. And the main thing that's happening here is the electrons are being used through a series of enzymes to convert ADP into ATP. Okay, water is made here, right? So one of the things we know as a product of cellular respiration is not only CO2, but also water comes out, right? If you don't believe me, go outside and breathe, right, on a cool day. You wouldn't see the CO2, but you certainly see that water vapor coming out. That's largely coming from this process. So what's going in? Electrons and ADP. What's coming out? Lots of ATP and water. Where is this taking place? Again, in the mitochondria. And essentially what is happening is you're creating a gradient of ions across this membrane. In other words, there's an unequal amount of ions across this membrane. And that unequal ions, if you look on the right-hand side of this diagram, are what drive ATP synthase, which is an enzyme that, as you might imagine, synthesizes or makes ATP. Okay, so you don't need to know all those different parts. You don't need to know the carriers. Each of those purple things are different enzyme complexes, but take a look at the broad, bigger picture, right? Hydrogen ions are going through the ATP synthase, and when they do so, when that gradient is crossed there, I'm sorry, when it's going through sort of the ATP there, the ATP synthase there, you can see the ATP being made. This is an aerobic process, needing oxygen. Okay, so finally, sort of an overall overview of this, and we'll put a couple links, to help supplement. But this really, if you studied only this picture, you'd have about 75–80% of what you need, right? So glycolysis, take a look at what it is. If we know that glucose is a 6-carbon sugar, that's what these 6 spheres of black are. It gets split, right, to lyse the the glucose, glycolysis, and you get these 2 3-carbon pyruvic acid. Okay, note where this is taking place, outside the mitochondria, in the cytoplasm. 2 ATP are made. The Krebs cycle, right, sort of represented here, is taking place inside the mitochondria, and this, again, is making a little bit of ATP. This is where CO2 comes out as a byproduct. But the main role of the Krebs cycle is to take electrons and pass them on to the next process, to the next series of enzymes embedded in this membrane. That is where the majority of your ATP comes from. Okay, so remember, ATP synthase is embedded in here, a lot of it. And when you have a gradient across these 2 different areas, right, which is this membrane is between them, that is the process with oxygen present that makes H2O, right, water as a byproduct, but mainly right here makes ATP. So ultimately, 1 glucose can be translated into 36 ATP if everything goes well and efficiently in this process. It doesn't always, but if everything goes well, you're converting that glucose and the bond energy in that glucose that you've eaten into this ATP, which can be used for all these different cellular processes, active transport, turning on and turning off channel proteins in the membrane. There's a lot of different things that it does for us. Very important. All right. Hopefully that made sense and you were able to get the notes down, and we'll see you soon.